Localization of proteins in the cell. Lecture 7. Experimental localization of proteins, number 2. In lecture number 6, we saw that one way of experimentally localizing proteins is to isolate the cell part and then identify the proteins in that cell part using mass spectrometry. But there's another way of doing it, and that is by using fluorescence labeling of the proteins. Now, fluorescence labeling of proteins is a fairly new method. What you do is that you label the protein with a fluorescent protein. So the protein that you want to study, you add a green fluorescent protein, which derives from a jellyfish. What you do is you make a DNA construct, that is an artificial gene, encoding the protein to be investigated with a fluorescent protein bound to the C-terminal. And the fluorescent protein is bound to the C-terminal to avoid interference with the N-terminal localization signal. Then you expand the DNA construct with a separate gene encoding a fluorescent marker for the potential cellular home of the protein, for instance, the mitochondrion. You insert the DNA into the cells and let them express both fluorescent proteins. So both the ones you want to investigate and the mitochondrial marker. Then you study the fluorescence of the cells to see if the localization of the two proteins coincide. If the two fluorescence coincides, then the protein you are studying is mitochondrial. The discovery and development of the GFPs was awarded the Nobel Prize in Chemistry in 2008. So it's quite a fairly recent technique. Okay, so here's an example, or rather 6 examples, because these are six mitochondrial proteins. The numbers are on the left. It doesn't really matter what proteins they are. And we have here used enhanced yellow fluorescent protein. So each one of these 6 proteins were labeled with enhanced yellow fluorescent protein fluorescence tag, and then co-expressed with the mitochondrial fluorescent marker in tobacco epidermal cells. So for each protein, that now show 3 images. At the left lane shows the EYFP fluorescence. The center lane shows the fluorescence of the mitochondrial marker, and the right lane shows an overlap between the 2 types of fluorescence, where the green color indicates the coincidence of the 2 probes. And if they coincide, well, since the marker shows mitochondria, then the green color will also show that they're both in the mitochondria. And you see there's a very good agreement between the EYFP fluorescence and the mitochondrial marker fluorescence. So all of these proteins, all 6 proteins, are mitochondrial. Well, that's nice. But we're still left with the problem of where in the mitochondria. And on these fluorescent images, you can see that the little dots are mitochondria. And the resolute, at the bottom, you see that the black bars are 12.5 micrometers. So a mitochondrion is really quite small here. And it's quite impossible to see where in the mitochondrion the fluorescence is located. But we have to do slightly better than that. So we have here a case story for a mitochondrial inner membrane protein. the protein, the mitochondrial calcium uniporter, MICU, or MICU, let's call it MICU, co-purifies with mitochondria. We know that. We also knew that it was a membrane protein catalyzing the movement of calcium ions across a membrane. So it was expected to be an integral protein in the inner mitochondrial membrane, not in the outer mitochondrial membrane, because it doesn't have calcium transporters, and definitely not in the two soluble phases, the intermembrane space and the matrix. So the question addressed by the experiments I'm going to show you was therefore, is it in the inner membrane of the mitochondria? Probably yes, but how to show it? Okay, this is quite complex. So let's just take it nice and slow. Fluorescence labeling of proteins, mitochondrial inner membrane protein, part A. In all cases, the cells are stained with a mitotracker orange. That's a small fluorescent molecule that goes into the mitochondrial matrix. So this is not a protein. So the cells are stained with mitotracker orange, which stains the matrix. On the left column, we have mitochondria transformed with MICU GFP, showing where the mycoprotein is found. In the center column, mitochondrial cells transformed with mito-GFP, a marker of the mitochondrial matrix. So we also have a protein marker in the mitochondrial matrix. In the center column of A. And the right column, mitochondria in cells transformed with NMT1 GFP, a marker of the mitochondrial outer membrane. Okay, so in all cases, the dots you see are actually mitochondria. That little bar at the left column of A at bottom is 1 micrometer. So all these mitochondria are 1 micrometer across. So the top row shows GFP fluorescence. The bottom row, a middle row shows mitotracker fluorescence. And the bottom row shows merged top and middle. And as you can see, there's very good overlap between the GFP, the mitotracker, and the mitotracker fluorescence, which is good. So they're all mitochondria. All the little green dots at the top. So, yes, it is mitochondrial. But that was where we started. Where in the mitochondria? So we look at part B. So the GFP and the mitotracker fluorescence is scanned across each of these individual mitochondria. Diameter about 1.5 micrometers. And then in B, it is plotted as a distance from the center of the mitochondria. Each of these little almost circles. Now in B, the left plot shows that the overlap between GFP and fluorescence and mitotracker fluorescence is not perfect. You can see that GFP appears to be slightly outside the matrix space. Remember, the mitotracker is found only in the matrix. And the mitotracker has a nice bell-shaped curve, whereas the GFP fluorescence looks like it has two peaks on either side of the matrix. In the center, there's perfect overlap between mito-GFP fluorescence and mitotracker fluorescence, which is what you would expect because the mito-GFP fluorescence was directed to the matrix. And the mitotracker is in the matrix, so they would be expected to, they are both in the matrix, so they would be expected to give the same pattern, and they do really nicely. B to the right, the overlap between NMT1 and GFP fluorescence, which is a marker of the outer mitochondrial membrane, is very poor. It's much worse than for MyCoo GFP. Okay. So now we look at part C. Modeling shows that the labeling of the inner mitochondrial membrane, the matrix, and the outer mitochondrial membrane would give precisely the patterns observed experimentally in part B. On the left, you see a two-peak pattern. In the middle, you see a bell shape. And on the right, you see a marked two peaks. So the conclusion is that the MyCoo GFP is clearly localized inside the outer mitochondrial membrane and around the edge of the matrix, in other words, in the inner mitochondrial membrane. Beautiful work, in my opinion. However, it has its limitations. Only some cell types can be transformed. That is, incorporate the artificial gene into their DNA and express the protein. The attachment of the GFP at the C-terminal end of the protein, target protein, might interfere with the correct sorting of the protein. And what's even worse is that it's very slow, as a new DNA construct has to be made for each protein, and a cell line must be transformed before the localization can be studied by fluorescence microscopy. All of that has to be done for each protein separately. So, the take-home message from lectures 6 and 7, experimental localization of proteins, is that subcellular protein localization can be accomplished by two methods. There's isolation of cell parts followed by identification of proteins by mass spectrometry. And there's fluorescence labeling of proteins. Both methods are time-consuming, and in many cases, neither method will give a clear answer. There are databases containing experimentally established subcellular localizations. Really quite extensive databases, SwissProt, NCBI, and for plants, SUBER, subcellular localization database for proteins. The other two also contain plant proteins. So because of the experimental localization of proteins is so time-consuming, there's a very real need for developing computerized methods. And that is the subject of another series of lectures in silico localization of proteins. Thank you for your attention.